Okay, we are recording, hopefully. So yesterday we left off talking about polysaccharides and glycogen. We talked about muscle. I think someone brought up the liver. That's where we store a lot of our glycogen. That's why livers are so dark and rich looking. Yeah, I'm recording. Yep. And so I know not all of us have opened up an animal and taken a look inside, but if you did look at a liver, it's a very dark, rich color, right? And then you think about the dark meat in turkey, it's really dark, right? It's dark meat. It has a lot of glycogen in it, and that's why, because of the glycogen. Okay. All right, so we've talked about starches. We've talked about glycogen. Cellulose is found in the cell walls of plants. That's why when you take a twig off a tree, you bend it, it goes snap, right? It's rigid, and we bend our elbow. We don't have that. We're an animal. We don't go snap. Unless you hit the bone, that's a different type of material. We'll talk about that later. Or if you've had anatomy, you already know. That's compact bone. So chitin is another one. And if you remember in anatomy, we say chitin is a lot different than cellulose. It's what shrooms or mushrooms are made of, right? You step on a mushroom, it doesn't go snap like a twig. It goes squish, right? I wonder if I got that in the recording. So I shouldn't be making fart noises in my device, right? Okay. All right. They're going to think something happened. We'll play that back and see what it sounds like before I publish it to YouTube. I don't know. I'm joking. It's okay. So chitin, we know, is the protein that, I'm sorry, not protein, the polysaccharide, I should say, that mushrooms are made of. That's the structural material in a mushroom or a fungus or a mold. And then there's this fun thing called peptidoglycan, all right? And this is found in the cell walls of bacteria. You guys probably remember talking in biology last year, the year before, about the different cellular structures of like eubacteria versus archaebacteria. And we'll get into that more later. Don't worry about that right now, but just understand that there's another polysaccharide called peptidoglycan. So it's a fun one to say. I always say, for some reason, peptidoglycagon, and I don't know why. It always comes out of my mouth like that. If I don't read it, like sound it out in my head as I do it, I always say that. Sounds like I'm adding a new ending to Pepto-Bismol when I do that. So cellulose, very similar to glycagon, or starch, I should say. These are the two ways that plants store energy in starches like potatoes, and we store energy in glycagon in our liver, glycogen. God, I just did peptidoglycagon with glycogen. Okay, so they are both polysaccharides, both long chains of glucose. But we are a plant, so we just have a different molecule, slightly different. Okay, so a potato is actually, think of it as a spare tire for a potato plant. So it's like a bulge of fat in a way, or that's not really fat. It's a bad analogy. But it's a place where that potato plant stores energy for the winter, right? Okay, and for times where it needs energy. So these are actually, potatoes are called, these things called tubers. It sounds like a tumor, but it's called a tuber, and that's where they store all the starch at. We store ours in this organ, not a tuber, called a liver. Okay? But we also have it in muscles as well. The next category we need to dive into are lipids. And we all know lipids are fat. How do you know? I think about the word liposuction. That word comes from lipid, right? And then I go, oh, well, lipid, liposucking. Okay, and that's what the word means. So if you know what liposuction is, you know, they cut a hole in your belly and they shove a garden hose up, hooked to a vacuum, and they suck all the fat out. It's lipid sucking. That's what the word means. Okay? So lipids are going to vary in structure too. They're large nonpolar molecules that are insoluble in water. So we know that lipids are hydrophobic, right? They're scared of water. Okay? Don't believe me, pour some gas on top. Don't do that. Don't believe me, pour oil on top of a glass of water. But if you think about an oil spill, that's a lipid, right? Okay? So an oil spill, you see them, they don't sink in the ocean, they float on the surface. You can actually light them on fire and get rid of them, right? So the function are they're long-term energy storage molecules for us. That's the way we use them. They're structural components. Okay, so they're part of our phospholipid bilayer. So we have these lipids that make up all of our cell membranes. And they also work with cellular communication and regulation, and I automatically think of the cell surface markers, the glycoproteins. Okay, I don't know if you remember those or not. And then they also serve for protection. So there's one organ, well, a couple organs in your body that are protected by fat. What are they? Anybody ever box? No. Anybody ever watch boxing? Yeah, like fighting. Yeah, or like who watches the Ultimate Fighting on TV? No. Shoot. Okay. So yeah, okay. So they always go for like if you drop in for a low shot, you don't go for the bony area, do you? You go for that soft area because it's a kidney shot, right? And you nail them in the kidneys. Because there's these special fats there. They're called prostaglandins that they release a lot of pain. They hurt somebody really bad. And so we have, I don't know how I got on that topic, but I went there. But fats are there to protect you, right? And so this area that doesn't have any bones in it has what there? Fat, okay? And if you fall, we often fall on our butt, and there's a pile of fat there, right? Okay, makes sense. So it's protection as well. Around your organs, there's often fat too. If you've had anatomy, you know that, right? You reach into a cat and you go to pull a kidney out and you grab a pile of fat, right? You guys remember that? Who's had anatomy? A few of you have. You reach in there and you go, where's the kidney? Well, you got to dig through that gooey stuff, that pile of fat, and then you pull it out, you know. What? Well, yeah, some of you had some, I don't know what you want to call them. Lean cats, yeah. What? I missed something, didn't I? No? Okay. All right, so the varieties of, that's a bad joke. I'm not going to tell that one. So the different varieties are fats, oils, phospholipids, steroids, and waxes. Those are the different varieties of fats. I always say fats should have been broken down into a few different categories, but they all have these fatty acids in some way, shape, or form, so we call them fats. And they're all lipids. So looking at the different categories of fats, we understand that fats are for long-term energy storage. Think of this as the butter or lard or that spare tire that old people try to get rid of, right? The stuff that makes, bacon taste really good. Then there's oils. Think of that as the vegetable oil. You can squeeze vegetables and get oil out of them. We don't have these types in us, okay? We have the more saturated fats. That's how we store ours, okay? But these are going to be found in plants and other seeds. Think about the oils on your shelf. There's no cow oil. There's no pig oil, although there is, but it's fat, right? Okay? You can buy lard and stuff like that and butter, but that's the fat. The oils are going to be like canola oil. That's a nut, right? And then there's peanut oil. That's another nut. And then there's vegetable oil, which is like all different sorts of vegetable seeds pressed and mixed into a blend. So I know a lot of us hadn't even thought about where the oil comes from that you use, but it comes from a plant. Okay? And then there's phospholipids. This is what our cell membrane is made of. How many fatty acids are in a phospholipid? 2, right? 2 fatty acid tails. Okay? And then we have steroids. These are components of the plasma membrane and also involve different sex hormones, testosterone if you're a boy, progesterone and estrogen if you are a girl. We'll talk more about those later in the year. We also use some of those in medicines. Anybody ever had albuterol when you have a cold? Inhaler type thing? Yeah? Or they give you the aspirator to breathe that stuff in? That is a steroid. How do I know it's a steroid? How many carbon rings do steroids have? Let's see if you remember. 4. If you see a structure with 4 carbon rings, we'll talk about this later, you know it's a steroid. Okay? If I give you a structure, well, 90% of the time, it's going to be a steroid. What? 4 different carbohydrates? What are we talking? 4 carbon rings. You know the ring structures we've been drawing and stuff? There will be 4 of those in all steroids. Okay? And then there will be functional groups attached at the end. Now, we'll talk more about that. Waxes are for protection and prevent water loss. The cuticle of plant surfaces, beeswax and earwax. We do have wax, okay? Our ears, various areas that we're trying to waterproof, we do have waxes. But often when I think of waxes, I'm thinking of plants and the coating on the leaves. Anybody ever grab a leaf and it feels like Tupperware a little bit? That's wax. So let's talk about all of these. First big category, the triglycerides. and these are for long-term energy storage. We call these fats and oils. There's different types of these. Their functions are for, again, long-term energy storage and insulation. They consist of one glycerol molecule linked to 3 fatty acids by dehydration synthesis. So if we see a fatty acid being added on, and I see 3 of them being added on by water being produced, I know that I'm looking at a triglyceride. So if you see a structure that has 3 fatty acid tails, 3 for the 3 fatty acids, glyceride, because there's an alcohol, sugar alcohol, glycerol involved, you know you're looking at a triglyceride. And I can be a fat or an oil. Fats are saturated. When I say saturated, what do I mean? What? Mostly single bonds, okay? Let me ask you guys a question. Let's see who knows this. When I say saturated, what would I be saturated with? Type it in a paradigm. When I say saturated, what would I be saturated with? Take a guess. The wrong ones are often the funny ones, and we don't know who you are, so it's okay. Take a guess. It's okay to be wrong. Unless it's inappropriate. Remember, this is going to be on YouTube later. Let's see what we end up with here. Yay, hydrogen is correct. Water is, well, part of water, right? That's 2 out of 3, right? Saturation. You're in here. Lipids, okay. Sugar. It's hydrogens, actually. So you guys, water. So I'm glad I asked that question, because when you go to a store and you look at the back of your package and you say, okay, what's there? And it says you have saturated and unsaturated. Well, saturated means I have more hydrogens bonded to the outside of my fat in that case, which makes it harder or easier to break down. What do you guys think? Harder. So you're not going to be able to easily metabolize that and get rid of it. So if it has more hydrogens, it's going to be a little harder to break down, and you're going to end up storing that generally as the fatty deposits that we think of as fat in our own body, or transfer it to them. The unsaturates are a little bit different. But the formation or the foundation of all fats are going to involve a glycerol. Okay? This is a basic glycerol molecule. Then we're going to have our 3 fatty acids that will attach to it. So if you see 3 fatty acids coming together, what's going to be made in this process? If I see on this glycerol, I have 3 spots where I could lose a hydrogen to a hydroxyl group on a fatty acid. If this happens 3 times, how many water molecules am I going to make? 3, but I'm going to make water, right? We don't care about how many, really. We just say, well, water's made, and there's 3 fatty acids. It must be a triglyceride. Okay, so if this happens, I go, okay, I make water, dehydration synthesis, okay, and in the end, I'm going to end up with my triglyceride. Now this is a bonded molecule. These can generally go both ways. Does this look saturated or unsaturated, this particular model? It looks pretty saturated with hydrogen, right? Okay, so here I'm looking at what's called a saturated fat. Now, the more double bonds I make, see how that one's double bonded? What happened when that double bonded? What didn't I need anymore? Hydrogen. So that would be what? Unsaturated. Okay. So fatty acids are either unsaturated or saturated. Unsaturated is where 1 or more double bonds between carbons occur, therefore reducing the amount of hydrogen bonds that occur on the outside, or hydrogens bonding to the outside, I should say. Don't want to mix that up with hydrogen bonding. So they tend to be a liquid at room temperature. Think of these as plant oils. That's like canola oil. That's the vegetable oil that's in your mom's baking cover at home, or dad's. It's the 21st century, right? I do most of the cooking in my house, so this is my oil in my house. Then we have saturated. There's no double bonds between carbons, and they tend to be solid at room temperature, like butter and lard. Not too many people cook with lard anymore, but that's just animal fat in a can. If you had well, I don't even know if your great-grandma, you guys are getting kind of young now, but your great-grandma might have had a can of this stuff up on the shelf that just sat there, and then they'd bring it out to bake or cook with. And eggs taste really good in bacon lard, but it's not so good for you, right? Yeah. I know it sounds gross until you like no one tells you you're eating it, and then you eat it, and it's like, gosh, that's really good. What's in that? Pile of bacon lard, pig fat, you know. You know, your tongue's tasting it, it knows, you know, even if you don't. All right, so taking a look at the different types, what does that mean? Well, we understand the top part. Through dehydration synthesis, I can make a triglyceride. Through hydrolysis, I can also do what? Break it down into the 3 fatty acids and the glycerol, right? That's pretty hard to do, though. Also seems how water and fats don't mix. So now once you've made a fat in your body, is that pretty tough to revert that process for two things that don't want to come together? Super hard, right? You actually got to create so much heat energy that you force them to bond, okay, which involves like working out, right, raising your temperature, getting your metabolism going. And really, the best way to, you know, reduce your fat is to not eat it, obviously, and not put it in your body so you don't have to break it down. So you got to have a certain amount of fat, though. I always say don't watch the amount of fat you eat, watch the type of fat you eat, right? That's what I always say. All right, so let's take a look at these two types. We understand this is not saturated, okay? I can see I have double bond. See the double bond, double bond, double bond? I'm missing what here? Hydrogens. The fact that I'm missing those hydrogens makes these a little less structural, if that makes sense to you. Because of that, they're liquids at room temperature, okay? These are very structured. See how rigid they are? They're all like, you know, standing at attention. 3 fatty acids all attached to that glycerol, lined up in a row. This is going to be very solid, or not very solid, more solid, I should say. And it's going to give us a structure because of this very organized, fully fulfilled hydrogen bonding that occurs all the way around my fatty acids. Everybody get that? Okay. You know, I didn't learn that till college. That was ridiculous. How did I get through high school without someone telling me that? That's how far education has come now. It really is. Heck, they hadn't even decoded the human genome when I was in school. Yeah, I know. Oh, okay, you got to bring that up. All right. Yeah. Yeah, touche. Yeah. Yep, it would have. Okay, so phospholipids. Looking at another type of fat, or I should say lipid. I don't want to say fat, because if I'm saying fat, I'm talking about a triglyceride, right? Okay. So looking at the structure of the cell membrane, we know it's similar to triglyceride. We can actually call it kind of a diglyceride in a way, but we don't. It consists of one glycerol molecule linked to 2 fatty acids. I wish they would have called it a diglyceride. That would have made much more sense in my mind, but they didn't. And a modified phosphate group. Now, the fatty acids are nonpolar and hydrophobic, which makes them unique in the fat world, right? They're both polar and nonpolar at the same time. The modified phosphate group is polar and is hydrophilic. So the phosphate end, oh, I'm really going to draw on some knowledge now. What is the overall charge of DNA? It's not neutral. It's negative, right? It has a charge on it, right? Where is that charge located? Oh, it's on the edge. Is it on the, and what's on the sides? There's the phosphate group and the sugar. So it's the phosphate group, right? Ah, okay. So if you're telling me phosphates will have a negative charge, that means they're going to be attracted to water, which makes them hydrophobic. Okay. Or philic, sorry. My brain's not working right. So, yeah, you get her now? Okay, so what I'm doing there is just trying to make a little connection between different things we're going to talk about this year. So the fact that phosphates are what makes DNA negative, it's also what makes our phosphate group attracted to water, all right? That electronegativity, we'll call it, all right? So because of that, that phosphate head is going to be drawn to water. Is water inside of the cell? Is water outside of the cell? Yes. So that means that the heads, you're going to need 2 of them, right? Are both going to go to in and out of the cell at the same time. And the tails are going to be where? In the middle of the membrane. I always say they tuck tail and hide because they're hydrophobic, scared of water. Can you imagine being scared of water and living in Michigan? You just have to drive like right down the middle. You know. What road is that? It's not 75, it's like 27. So, why does my brain go there? What? It's not right. So when you form plasma membrane, can you imagine like a phospholipid or a lipid on a ferry, just, you know, shaking, water everywhere? So in water, phospholipids aggregate to form lipid bilayers. We know that. So polar phosphate heads are oriented towards the water. So we have two layers here. Both heads are on the outside. The hydrophobic tails, the phospholipid fatty acid tails, are going to hide on the inside. How many tails are in each phospholipid? 2. Remember, we could call a phospholipid a diglycerol. So nonpolar fatty acid tails are oriented away from water, and nonpolar fatty acid tails form a hydrophobic core or middle of the membrane. Tuck tail and hide. So it looks something like this. Here's my 2 fatty acid tails. They're very bond happy, we'll say. Okay, that means they're kind of neutral. They don't have that electronegativity that my head does. If I had another tail that would have bonded here, I would have had a triglyceride, but I don't. Because of that, I have a little instability in my molecule. I have this phosphate that, again, pulls on those oxygen groups, and we know oxygen is electronegative, which is going to make this end attracted to water, and is going to make it hydrophilic, right? Okay. So on the cell membrane, in the middle, we have our tails, and on the outside, we have our heads. Makes sense, because there's water in and out of the cell. We good there? This should have been kind of conceptual knowledge that you came into class with. Just give you a little more detail now of why, right? So looking at steroids, those are 4 fused carbon rings. If you see 4 carbon rings all together fused, you know you're looking at some type of steroid. In this class, we'll say. I don't care about chemistry class. In this class, you see 4 carbon rings fused together. It is a steroid. So it's composed of 4 fused carbon rings. There are various functional groups attached to the carbon skeleton. There's all different sorts of steroids out there. There's all sorts of synthetic steroids that are made, and they all have different functional groups. But the one thing they have in common is those 4 carbon rings. They are components of the animal cell membrane. Who's heard of the word cholesterol? Steroid, okay? So you have good cholesterol and you have bad cholesterol, right? HDL and LDL. So there's different types of functional groups on those 2 different types of cholesterols. And they're all part of our cell membrane. Do you suppose good cholesterol is more or less pliable than bad cholesterol? More pliable, right? The bad cholesterol is kind of rigid. It doesn't flex. It doesn't sort of, imagine a sheet that has way too much starch on it, and you go to like fluff the sheet and it's like a board. Okay? That's bad cholesterol if it's part of your cell membrane. It's like adding starch to it. Good cholesterol would be like adding fabric softener, right? And it just makes it all fluffy and perfect and everything flows, and right? You get the idea though? Because one causes like hardening of the arteries. That makes sense now, right? And the other one does not. So cholesterol is one. Testosterone, that's the male sex hormone, causes all the problems in the world, right? Maybe? I'm just kidding. But seriously, it kind of does. But that's just because it's getting mad at estrogen, because you know there's this other. No, I'm kidding. But seriously, these are the 2 sex hormones, right? We understand that males have testosterone, females have estrogen, and that's why we're boys and girls. Okay. If I asked you where testosterone is made, you'd probably tell me testicles. If I asked you where estrogen is made, you'd probably tell me ovaries, right? Those are the 2 gonads. Okay. So cholesterol is the precursor molecule for several other steroids, which means often a cholesterol can develop into other types of steroids. Can you turn your bad cholesterol into good cholesterol? Change what you eat, right? So looking at the diversity of steroid, I don't really want you to focus on trying to identify the different types at this point, but what I want you to do is understand that if you see Uno, dos, tres, cuatro. That's all I can do in Spanish. Or un, deux, trois, quatre. Or 1, 2, 3, 4. Fused carbon rings. You know you're looking at a steroid. Where's the functional group? It's the thing that is different, right? So there's the functional group, okay? That's not, but this is right here. You understand, okay? Look at him in his testosterone. Look at him all puffed up. No testosterone. Looks like a good thing until it starts a war, right? And then we got a problem. Then waxes. Waxes are special lipids, okay? They're long-chain fatty acids bonded to a long-chain alcohol, kind of a really boring long molecule, but they're very structured. What do we know about very structured things? They're very what? Rigid and solid. So solid that and they're hydrophobic as well because they're all happily bonded, so they are going to repel water, okay? So they're solid at room temperature, they're waterproof, resistant to degradation, and they function to protect. Looking at earwax, I'm not going to have you pick your earwax out and look at it. You guys can do that in the morning. Plant cuticle, we understand that plants have this substance that keeps their leaves dry, and beeswax would fall under this category as well. That's what keeps bacterial infections and things like that out of their hives. Beeswax is really good on your boots. Anybody use beeswax on their boots? Waterproof them? Yeah. Either beeswax or mink oil, those are like the two main ones. Beeswax is good because it doesn't really break down like mink oil does. Right? In case you got to waterproof your boots later. Then there's a lot of synthetic products out there too, right? Oh, the last category, proteins. Okay. So proteins we know are polymers, those large molecules made of amino acids, right? They're linked together to form peptide bonds. If I ask you what type of bond holds amino acids together, you tell me a peptide. If I have two amino acids held together by one peptide bond, that is called a dipeptide, right? Okay? So when I refer to just one bond, one peptide bond, that is a dipeptide. If I put more than two together, then it is a polypeptide. So two or more amino acids joined together are called peptides. Long chains of amino acids joined together are called polypeptides. Are all polypeptides proteins? No. Are all proteins polypeptides? Yes. So a protein is a polypeptide that is folded into a particular shape and has function. I'm going to ask you a question real quick once you get back. Take your break.